Welcome to our third annual Graduate Research Symposium. My name is Emory Woodard, and I'm Dean of Graduate Studies in the best college at Villanova, the College of Liberal Arts and Sciences, if I may say so myself. We're gathered today to both hear and celebrate the fine work of our graduate students in the college, that best college in the university. I'm not hearing any celebration of that. All right, that's better, that's better. You're about to see just a small sample of some of the wonderful work produced by our students from across the college, some of whom received 2021 Summer Research Fellowships, and one student who submitted a project just for presentation at this symposium. Let me tell you a little bit more about the Summer Research Fellowship Program. It's a funding initiative to support students who wish to stay engaged in their research over the summer. It's awarded to folks who are in good academic standing in either a master's or doctoral program in the college and are eligible to receive a stipend of $3,000 to support their scholarly efforts. These awards are given on a competitive basis. A faculty selection committee evaluates submissions for their feasibility, clarity, and potential for scholarly contribution. And this year, I am pleased to announce that we were able to support 25 such awards. Now, in the fall of 2019, through a collaboration with the Center for Research and Fellowships, we hosted our first symposium in which awardees from the summer before made posters or oral presentations of their work in this very Connelly Center. COVID-19 did not allow for such an event last year. So instead of the cozy spaces in this fine building, we hosted a Zoom event through which we heard from our resilient students who, despite the interruption of COVID-19, carried out their exciting work. Beginning last year and continuing this year, we have asked our presenters to use the 3MT style to share their work. 3MT, or 3 Minute Thesis, is an international research presentation competition in which graduate students from around the world engage in the important professional development exercise of communicating their scholarship in 3 minutes or less to a non-specialist audience in as compelling a manner as possible. These competitions reinforce the notion that in order for our work to be most impactful, we need to be able to effectively convey it to others in a way that they can understand. That is when the work that we do in the academy can truly make a difference in the everyday lives of those outside of it. As an added bonus to reward those who master these skills, the competitions include cash prizes. All of the presenters today will automatically be considered for our spring competition, and that spring competition seems to be moving toward a university-wide event. And I'll tell you more about that later. And it will be hosted in the plush Mullen Performing Arts Center. In addition to these presentations from the stage, some of the fine work of our students will be on exhibit in a poster session following this part of the symposium. Now, the delineation between poster and oral presentations was based largely on what a review team in my office thought would be the most effective modality for conveying the important work of our students. In fact, 2 of our highest rated projects will be presented as posters, and you certainly don't want to miss them in the session that follows these oral presentations. Also, during those poster sessions, we're going to be serving some snacks and awarding not 1, but 2 pairs of Villanova men's basketball tickets for the game on this upcoming Tuesday. So for now, we're going to enjoy brief 3-minute distillations of the work of a sample of our graduate students, after which we'll have a very brief Q&A period of about 2 minutes. Again, we've got a lot of presentations. We tried to fit as many in as we possibly could, so we're going to be moving things along at a steady clip. But I do want you to engage as time permits, because truly, I have thoroughly enjoyed the presentations that I was able to review in our evaluation process, and I think you're going to enjoy them as well and be engaged by them, energized by them. So if you've got a question, be the first to get your hand up so that we can get it in. All right, so right now I just want to cover some brief housekeeping items so that we can make the most of our time together, let you know about some upcoming opportunities, and then pay a few debts of gratitude. In terms of housekeeping items, please note that we are recording this event for our posterity. Please refrain from moving around during the 3-minute presentations. Use the Q&A period and the transitions between presentations if you need to take a biology break or attend to other needs. One of the things that you may want to do during the interludes is make sure that you have completed an entry in our VU men's basketball ticket raffle. Again, we have 2 pairs of tickets to the Tuesday game against Howard University, and they will go to just the folks in attendance. So make sure that you fill out an entry. All faculty, staff, and student attendees are eligible to win, and we're going to do that drawing right at the end of the poster session. Now, in terms of upcoming research opportunities, particularly if today encourages you to get more actively involved in your scholarship, we have Concept, the interdisciplinary journal of the Graduate Division of the College of Liberal Arts and Sciences. I am going to invite John Kurtz, the managing editor, to tell you more about that at the end of the program. We are pleased to announce not 1, but 2 competitions for graduate student research fellowships for summer 2022. Beginning next summer, we will have separate competitions for master's and doctoral students. The deadline for master's student applications is February 1st. The deadline for doctoral students will be March 1st. Any graduate student in the College of Liberal Arts and Sciences may apply for a summer stipend to support their scholarly efforts. And of course, again, the awards are given on a competitive basis in order to support the fine work of our students. Finally, I want to give a quick plug to our 2022 3MT competition. It will be an open competition for our 2022 representative to the regional 3MT competition. Our local competition will be on Friday, February 18th, and it will be hosted in the lovely Topper Theater in the Mullen Performing Arts Center. Again, it's looking like our friends in engineering, the College of Nursing, the Villanova School of Business will be joining us in this event. There will be a cash prize, and perhaps since they're joining us, it will be even a bigger cash prize. But do attend to your email to learn about that opportunity as those details are finalized. All right, the last thing I want to do before we hear from our first presenter is pay a few debts of gratitude. One of the first debts of gratitude that I need to pay is to our 2021 Summer Research Fellowship Selection Committee. Is Dr. Anil Bamazai here? He is one of our committee members, and he hails from the biology department. Dr. Sarah Von Brackman from philosophy. I saw Dr. Lynn Hartnett from history come in. Dr. Daniel Kraut from chemistry. Dr. Tom Kizak from communication. Dr. Peter Spitaler from theology and religious studies. And Dr. Joe Toscano from psychological and brain sciences. I also want to thank all of the graduate faculty sponsors, some of whom are here and will be presenting their students. These folks mentored the students through their projects, helping to nurture those projects to their successful completion. Charles A. Walsh funded one of our fellowships that went to one of the highest rated proposals by our selection committee and is due our thanks. Finally, I really want to thank my team in the Graduate Studies Office. Specifically, I think I owe a huge debt of gratitude to Brooke Erdman. She is responsible for organizing the fine event. And I thank all of you for joining us. All right, without further ado, okay, let's clap. Without further ado, I am pleased to present our first presenter, Shane McFoy from biology. Shane will be presenting Social Networks of Mixed Species Chickadee Flocks Change with Food Accessibility. Shane, take it away.
Awesome. All right, how's this? Can everyone hear me? Perfect, awesome. Cool, all right, so I kind of want you guys to think about your friend groups, right? they influence you in some way or another. You probably have a couple different friend groups that you hang out with, all of whom have different interests, right? And you might share some interests with them, or you might not. And in today's day and age, you probably connect with these friends through some form of social media, right? Whether it's Twitter, Instagram, Facebook is a huge one, obviously. You're connected to your direct friends, but you're also connected to your friends' friends, right? There's this whole network, this whole social network going on. And though the interaction with those indirect friends isn't as intense as it is with your direct friend, they're probably still influencing you somehow, right? You're seeing what they're posting on Facebook, the discussions that they're in, their likes, their dislikes, etc. And, altogether, these people have a big influence on your life. If a friend tells you that they got food poisoning at a restaurant, you're probably not going to go there, right? You don't want to get sick. You saw that they were puking their guts out. That's disgusting. So what if I told you that, in this respect of social groups, you're not so different from birds, right? People say birds aren't real, but they are. So birds also form social groups. So as ornithologists, we see that certain species form what we call mixed species flocks. So it's a couple different species that are kind of living together, flocking together, and there's a bunch of benefits that come with that. They have better predator avoidance, increased information, relay and transfer, and it also increases their learning. So, it's essentially, we're able to create these social networks, as you can see. Oh, my slide. I should probably put that up. Uh-oh.
Just use the arrow.
Cool. We create these social networks with the chickadees. You can see who's hanging out with who. So the circles are called nodes, and that's an individual bird within this mixed species flock. And I'm specifically looking at Carolina hybrid and black-capped chickadees and how they're interacting with each other at our feeder arrays. As part of my larger study, my thesis study, I'm collecting the social interaction data. We go out, we collect the birds, and we give them, essentially, it's an easy pass, a little RFID tag that goes around their leg. And when they come to my feeders, it records the date, the time, and their ID number. So we're able to see which birds are interacting with each other, at the feeders, and it's really cool. So the nodes are the circles, those are individual birds, and the lines are called edges. And the thicker the line, the more intense the interaction is between those two birds. So you can see we have some pretty thick lines up there. Those birds are interacting quite often. And with that data that I'm collecting from the feeders, we create these social networks. So we get these webs. And my thesis is looking at specifically cognition, but the social network data that I'm collecting creates these webs, but the really important thing is that you can see these communities that are forming. The feeders, they have 3 modes that they put them in. Open mode, where it's a mechanical door that remains open, and any bird in the area can come, regardless of if they're pit tagged or not. The next one is all access mode, so all pit tagged birds, when they come to the feeder, the door will actually open for them, and they can get the seed out. And then the final mode is target mode. So the birds that are pit tagged, they're only allowed access to a specific feeder if their name is on a VIP list that's installed on the board. And throughout these 3 stages, you can see that, I'll tell you that the food is cut off from the birds. If you're not on that list, you're not getting to eat the seed inside, right? So a trend that I was kind of seeing when looking at this data and analyzing it was that the social structures of these birds, the Carolina, the hybrid, and the black-capped chickadees, they change. So yes, we have 2 distinct groups. You can see there's a blue group and a pink group. But in each mode, different birds are interacting with each other. And these 2 different communities are kind of forming, which is really neat. So overall, the data is kind of showing us how flexible, or a term that we use is plastic, learning and cognition in these birds is. So Yeah, they're kind of smart. They kind of figure things out. But that's, I believe, everything that I've got for you guys. So thank you very much.
All right, we have 30 seconds for one question. 30 seconds. Any questions?
Hit me. Yes.
Why chickadees? Are they known to be particularly social, or would you expect to see different behaviors from different kinds of birds?
Yes, that's a good question. Chickadees are they're social birds. They have these mixed species flocks. Everyone kind of has a different role. They're satellite species, and they kind of recruit people or other birds into their flocks. But what I'm specifically interested with my thesis is we have black-capped chickadees, that are up north more so in their range, and Carolina chickadees down south, and they meet in this place called the hybrid zone. And they meet and they have these hybrid babies. So we're kind of looking at how the cognition of these hybrids is compared to their non-admixed parental species. So this was kind of just a little part of what I'm doing, but that's the reason I'm looking at chickadees, is because, essentially, how smart are these babies, and then how does that affect not just their individual survival rates, but also the entire, fitness of the whole species, if that helps. Sweet.
Great. Well, thank you so much.
You've surely seen them on a stroll through the park on a pleasant summer day. Chances are they didn't catch your attention, even though they are genetically altered mutants. They might even be in your front yard right now, and in fact, you've likely grown them yourself. I'm talking about marigolds, a plant that is ubiquitous to the point of banality. But it has been said that the familiar is not necessarily the known. I believe this is true of many of the living organisms we encounter on a regular basis, especially plants in the wild. Our lived environments are increasingly human-made, but even our most urbanized spaces are home to plants. These plants, either through sheer adaptive will or with the help of human intervention, both planned and unplanned, share our urban spaces. This is just one aspect of a relatively new area of study called urban environmental history. I'm particularly interested in the ways that human networks, our cultures, economies, and technologies have shaped our relationship to plants as they and we have moved across the globe. Like humans, plants have been immigrants, subject to the trials of assimilation or rejection in their new cultural environments. The lowly marigold is actually a well-traveled cosmopolitan flower. Its roots can be traced to the Aztecs, who grew the plant for ceremonial use. Spaniards brought the plant back to Europe as part of the plunder from American empires. The marigolds then followed missionaries to North Africa and then eastward as it was adopted as an essential element of South Asian cultural celebrations. The Puritans brought the plant back to the Americas. My study focuses on attempts to popularize the marigold in 20th century America. Much credit has rightly been given to one man, David Burpee, owner of the hugely successful Burpee Seed Company, located just 40 miles outside of Philadelphia. A shrewd businessman and innovator, Burpee used the latest breakthroughs in chemistry and atomic science to force genetic mutations. From these mutations, new plant varieties, such as the X-ray twins, were marketed to Americans, enthralled with hope that technology would ensure a better future. Burpee hoped that one day he would create marigolds in red, white, and blue that would grace the gardens of Washington, D.C., a place he had keen interest in, and also that his seeds would be the first seeds sent to the moon. Interestingly, David Burpee sought to associate his new marigold varieties with American diplomacy. Burpee called his plants friendship flowers and American marigolds. For publicity, he sent baskets of his friendship flowers to English Queen Mary, the West German Chancellor, and even the Emperor of Japan. But he didn't stop there in his quest to make the marigold as American as apple pie. He lobbied Congress and incited senators to debate naming the marigold the national flower. Ultimately, despite his extensive efforts, the marigold lost out to the rose. Though it never achieved the lofty, iconic status of the eagle, the marigold certainly assimilated into gardens across the nation. As a microhistory, the story of the marigold offers an entry point to debates about the tangled relationship between culture and nature, or human-made and biologic domains. It asks us to consider the socially constructed ideal of nature itself and our ever-changing relationship to it as we contemplate our place in the Anthropocene. Thank you.
So thank you for that brief introduction. Like you said, my name is Hannah Kessler, and I'm a second-year master's student in the O'Reilly Laboratory. Our research primarily focuses on synthesizing potential antibiotics for treatment of antibiotic-resistant infections, and my work is specific to methicillin-resistant Staphylococcus aureus, or MRSA. Even though we already have great antibiotics on the market, bacteria have started to become resistant to these types of treatments. In 2019, there were 2.8 million antibiotic-resistant infections, and 323,700 were related to MRSA, causing more than 10,000 fatalities. Therefore, new medications for bacterial infections are urgently needed. Emicin is a functionalized dihydropyrimidine compound that was discovered by the Spring Group in 2008, and it was shown to successfully inhibit the growth of MRSA. It is believed to work through dihydrofolate reductase inhibition and is a promising lead for further development of antibacterial agents. Compared to the many other classes of antibiotics, such as the 34 approved beta-lactam antibiotics, all working through the same mechanism, trimethoprim is the only FDA-approved antibiotic that works through inhibiting dihydrofolate reductase, making it an underutilized antibiotic target. We developed a library of functionalized dihydropyrimidines by making small changes to the molecule. By making these changes to the compound, we are analyzing how it will bind to the target. For example, if we add or remove a group of atoms to the compound, it may interact with the enzyme more or less favorably, which will impact its potency among other properties. We're also studying the bioactivity of these analogs by using methicillin-sensitive Staphylococcus aureus and MRSA strains that were hospital and community acquired. Some of our compounds have shown inhibition as low as 8 micrograms per milliliter, and we're looking to test at lower concentrations in the near future. Lastly, we wanted to confirm the mechanism of action of our compounds as we hope they inhibit the underutilized antibiotic target, dihydrofolate reductase. Toward this end, enzyme kinetic assays are being used to investigate the ability of our compounds to inhibit their presumed target. Preliminary studies indicated our compounds provided modest inhibition of the enzyme, but we are still testing different variables to make this assay more reproducible and will be continuing throughout the fall and spring semester. I would finally like to thank the College of Liberal Arts and Sciences for the funding that they provided this summer to carry out research. Thank you.
And thank you, Hannah, for those kind words. I really appreciate that. Questions for Hannah? Yes, Emily.
Is there a danger to studying this in the lab? Like, how do you prevent? This is a layperson's question, but.
No, there's no danger to studying this. No, we use a biosafety cabinet, so that kind of keeps everything sterile in our lab. So it's pretty safe.
That's a good thing.
Yeah.
All right, thank you so much,
you.
Hannah.
Thank you. The police are something of a fuzzy point in secular society. Reverence for the police is seen as the basis of any community's common life. At the same time, the police exemplify brazen patterns of deceit and domination. I've been investigating how cultural memory and imagination around the police are formed, developed, and transmitted, focusing at the intersection of law and religion. My thesis is that a critical focus on police power is a crucial part of any larger project of diagnosing social maladies, and that this focus requires a fundamental challenge to that power. Behind me on this slide is a clip from a local news agency, the Catholic Standard and Times, in which a pope pays high tribute to a group of policemen in Italy, urging them to act as friends, counselors, and helpers, rather than as representatives of oppression. The journalist team chose for the featured image here, you can see an officer directing traffic at a particularly busy intersection in Philadelphia. The image labeled as typical of policemen all over the world. In his talk here, Pius describes police work as on behalf of the common welfare, encouraging them to be good members of their community and not bad, stressing the importance in what he calls the dignity of their office. He stresses that police have been given, within certain limits, the power to watch carefully, to advise, to rebuke, and even to impose fines. Many of these instructions seem generalizable to a wider population. If we look at the bottom left, we see the police officer's prayer to St. Michael, heaven's glorious commissioner of police, praising the achievement of his having neatly and successfully cleared God's premises of all its undesirables. What was happening with the police in Philadelphia around this time? A contemporary piece in the Journal of Urban History coined the term dirty work to describe how police in Philadelphia were organizing against accountability, then in the form of a mostly toothless civilian review board, and developing the power of their own union in concert with ecclesial authorities. Earlier in that decade, a new city charter promised a new era of reform. While at the same time, a cast of characters, including Paul Robeson and others, jetted to Paris to accuse the United States of genocide, noting in particular the actions of its police, including specific Philadelphia incidents, such as when Charles Fletcher was slain by patrolman Manus McGettigan, who had claimed he shot after receiving a call about a prowler, or Raymond Kauser, who was walking down Montrose Street in Philadelphia when eyewitnesses saw patrolman Frank Kakuro stalking him with a drawn revolver. Kakuro fired, Kauser staggered, wounded. The patrolman fired 3 more shots. Reading Simone Weil, in particular her inquiries regarding the nature of repression, science, and work, has led me to believe this is all worth more careful study, tying together the ways we cannot yet read nor understand with the police who are stealing, killing, and destroying that which in other ways we might like to love. Thank you.
All right, thank you very much, Joe. All right, have you ever wondered why some conversations seem to go perfectly and others do not? I frequently have the same thought. Whether it's simple miscommunication or a laggy Zoom call, some conversations we have in our day-to-day lives seem to go astray. Now, this may be fairly innocuous for mundane conversations, like talking about your morning commute, but for more serious conversations, this can be more problematic. Specifically, I'm talking about goal-directed conversations. Those where we are not talking for idle chit-chat, but we are attempting to convey a specific piece of information to another party, and it's important that that other party comes away from the same conversation with the same understanding as our own. For example, a job interview is a goal-directed conversation. It's important that an interviewee and future employer share a mutual understanding of the conversation at hand, or else the interviewee may not get the job and the conversation becomes meaningless. This means that miscommunication is more detrimental to goal-directed conversations than to casual conversations. This process of establishing a mutual understanding of the conversation at hand is referred to as common ground in the world of psycholinguistics. One variable that impacts how we develop common ground is theory of mind. Theory of mind is the unique ability in humans to represent the mental state, thoughts, or emotions of another human while simultaneously maintaining our own representations. Why might this be beneficial for a goal-directed conversation and establishing common ground? Well, when having a goal-directed conversation, it's important to keep in mind what your conversational partner both knows and does not know. Additionally, should they say something incorrect over the course of the conversation, it's important to understand why they held that incorrect belief in the first place, and then explain the correction to them. This brings me to my summer fellowship. For my summer fellowship, I researched the question, what is the impact of modeling mental states on goal-directed conversations? To do this, I created 2 groups, an experimental and a control. There were 6 pairs of participants in each group for a total of 12 pairs of participants, 24 participants total. The experimental group was primed with a theory of mind inventory aimed with the intention of activating theory of mind cognition to make them more cognizant of their partner's thoughts. The control group received a similar prime that only assessed content and did not aim to activate any theory of mind cognition. After receiving these primes, participants solved 3 Lego puzzles. For these Lego puzzles, participants were able to see their own workspace, but not their partner's. One participant was in charge of directing the other participant on how to build an exact replica of the model in front of them. You can see on my PowerPoint our laboratory setup, as well as examples of these models completed flawlessly and with error, we hypothesized that participants who received the theory of mind prime would make fewer errors building these Lego models than those that did not. The results show just that. You can see on the PowerPoint slide, in green, the experimental group, and red, the control group. The experimental group not only made fewer errors building the models, but actually took less time to do so than the control group. This data has proven invaluable towards work on my thesis here at Villanova. Thank you very much for your time, and I'm happy to answer any questions.
Very convincing evidence. Questions about the theory of mind?
Yes?
Is there something to be said for the fact that some people might just have trouble building a model?
That's a great question, and the world of statistics would say that if someone is very good at building a model in one group, we should, with enough data, get an equal number of participants who are good at building those models in the other group. So it's a very valid point, but we hope that the world of statistics would say that that would cancel out from randomized assignment.
Well said, well said. All right. Samuel, thank you very much.
Thank you very much.
So at this moment, there is a huge and primarily ignored problem affecting millions of people. The problem is this. Mental health practitioners want to help autistic people, and autistic people want help from mental health practitioners. But what autistic people need from their mental health practitioner and what they receive are so totally different that often more harm is done than good. The crux of this problem lies in the mental health field's conceptualization of autistic people as less than so-called normal people. But normal is a pretty subjective concept, usually defined by whoever the predominant group is at the time. And studies show that autistic people don't want to be normal. They view their autism as a value-neutral and integral part of their identity. They don't want to get rid of their autism. They just want access to the same quality of life and mental health care that non-autistic people get. I was diagnosed as being on the gray area of the autism spectrum when I was 19. This experience launched me into a vibrant autism community. I first integrated into the community by reading autistic-run magazines. I noticed an overwhelming amount of discussion about stigmatizing experiences with mental health practitioners. From articles about people's therapists casually comparing them to apes and robots, to articles about people attempting suicide at the age of 14 to get out of anti-autistic behavior training. The saddest part was, as a mental health counselor myself, I knew these practitioners likely thought they were doing the right thing. They were almost always following the predominant treatment protocol. But their treatments wound up having two major negative effects. The first was damage to their relationship with the autistic client, which is the single most important factor of therapeutic success. And the second was corrosion of the autistic client's self-esteem, which is correlated to the astoundingly high suicide rates of this population. My work with autistic adults is critical for three reasons. One, this research provides them a platform to advocate for the cessation of techniques that they view as oppressive and stigmatizing. Two, it challenges the ethics and efficacy of the predominant method of autism conceptualization. And three, it generates practical suggestions for how to far better serve this population. A good place to start is to include autistic voices in decisions about their treatment. So much harm has been done by forcing a square peg into a round hole. What can we become when we are no longer the subject of psychology's hammer? Thank you.
Wow. Very nicely done. Questions for Zoe? Yes.
Do you think this is like unique to adults with autism, or do you think like the same issue is negatively impacting kids when they're getting treatment?
That's a great question. I specifically wanted to look at adults with autism for issues relating to feasibility with the study and IRB protocols and protections, but also because autistic adults are very much forgotten about by the system. There's often this huge push when you're a toddler to put you in all of these treatments that are going to get rid of your autism, to shape the course of your life, and then many autistic adults, studies show, feel like the system just gives up on them when they're adults. They just say, okay, well, we failed with you, and so you're on your own. And so much of the research out there about autistic adults is not even with autistic adults. It's asking like their parents or their healthcare provider to speak for them. So I really wanted to give this community a voice in my research. Thank you for the question.
Okay, so many of us in industrialized nations, we interact with a variety of computer and smartphone interfaces nearly every day. They help us get to work on time, order our dinner, take photos, and among other things, connect with others. Now, no doubt this tendency started with the proliferation of personal computers, but with the widespread adoption of the smartphone, especially the iPhone, and its appeal to attractive and intuitive design, our interaction with user interfaces has become nearly inescapable. Parents and grandparents never tire of reminding us of how younger generations are quote-unquote glued to their phones, yet those very same parents and grandparents can be found awkwardly scrolling through Facebook or some other app. Now, the immense benefits and pleasures of these devices notwithstanding, my work argues that everyday interactions on tantalizing user interfaces threatens our practical rationality, by which I understand our reasoning about our own desires and our capacity to deliberate both with ourselves and with others. In the dissertation, I intend to draw upon Aristotle and contemporary philosophers inspired by Aristotle in order to develop this notion of practical rationality and a philosophical framework to evaluate user interface design and experience. Now, within the industry of user interface design and experience, designers have been increasingly concerned with moving from designing for users and instead opting for what they call human-centered design. But a clear understanding of the ethical foundations of what they mean by human-centered design is still at a nascent stage. Now, in philosophy, we have been rightly concerned with the ethics of artificial intelligence, the ethics of surveillance, and the ethics of algorithms, but very few philosophers have examined the user interfaces themselves which these devices and algorithms rely on. It is the interfaces which are attractive and keep us using the apps, not simply the complex code which produces algorithms. Now, I hope to fill this gap in both industry and philosophic context, and I believe the time is ripe to do so, as we have seen with recent developments of whistleblowing at Facebook, as well as congressional concerns over dark patterns, which are tricks in user interface design that nudge users into making decisions which benefit company profits over the person using the product. Finally, in the dissertation, I argue when so much of our social and personal activity is mediated by predetermined, limited, and simplistic interfaces, we are liable to treat what's left of the offline world as if it were equally limited, simplistic, and predetermined. Ultimately, I worry that the habituation created by user interfaces will further entrench a culture of immediate and easy consumption over the complex, difficult, and at times boring reality that we live with. Thank you.
Questions for Christopher? Questions? Yes, Dr. Hartnett.
So thank you. So would you say that the influence, which has a greater influence, the qualitative or the quantitative in terms of how many things that we're seeing, or the nature of the interface that we are seeing?
Great question. It's hard to pick either. Now, I tend to think that the presentation is crucial. I mean, I think of it as like if you were served a dish that was wildly unattractive, even though it tasted great, you probably wouldn't go for it, or less people would. And so I think first and foremost, you need the front end part. You need to make it look good before we end up spending as much time consuming so much information.
All right, thank you, Christopher.
Thank you.
Thanks, Steve. So think back to the last snowstorm. Do you remember plow trucks spreading road salt behind them as they push snow to the side of the road? Have you ever thought about the impacts that road salting has on our environment? I personally never thought much about it. I just thought of these trucks as a winter staple. However, my perception of road salt changed when I started researching the water quality of rivers and streams in southeastern Pennsylvania. These trucks spread road salt, which is composed of sodium and chloride. While road salt prevents snow from sticking to the road surface and improves the safety for travelers, once it is applied to the roads, road salt can enter the nearby rivers and streams. This is concerning and can negatively impact both humans and freshwater organisms. First, our local rivers and streams are sources of drinking water to tens of thousands of people in southeastern Pennsylvania. Individuals who are following a low-sodium diet for the risk of elevated blood pressure can be subject to spikes of sodium in their drinking water during the winter months. Chloride has also been shown to be toxic to freshwater organisms, with the EPA setting thresholds in which the chloride concentration should not exceed to avoid negative effects. Previous studies have shown that sodium and chloride concentrations are increasing over time in streams throughout the world, and there is a positive relationship when watersheds are more urban and contain more impervious surfaces, such as roads, bridges, and parking lots. These trends are especially prevalent in regions that apply road salt during winter months. My research focuses on the long-term trends of sodium and chloride concentrations for 6 watersheds in southeastern Pennsylvania. I evaluated these trends in relationship to the amount of total watershed impervious surface cover, and I hypothesized that as the amount of impervious surface cover increases in each of the watersheds, the chloride and sodium concentrations will also increase. I found that impervious surface cover and chloride and sodium concentrations are increasing over time in all 6 study streams from 2005 until 2019. The streams with the greatest increases in impervious surface cover also had the greatest increases in the average chloride concentration over the study period. Based on my results, if current trends continue, the average chloride concentration in all 6 study streams will exceed the EPA threshold value by the end of this century. In 2019, the average sodium concentration in all 6 of my study streams exceeded the US EPA recommended threshold value for individuals following a low sodium diet. In 3 of my study watersheds, the average sodium concentration was more than double this threshold. Road salting is clearly having a negative effect on the water quality of rivers and streams in southeastern Pennsylvania. However, my study also provides guidance from a smart growth perspective on limiting future expansion of impervious surfaces in watersheds which serve as a drinking water source. So next time it snows and you are shoveling your driveway, think twice before applying salt.
Definitely think twice. It not only has these deleterious effects on the water supply, but it messes up good shoes. Any questions? Yes, right here.
Do our water filtration systems not help this problem?
Oh, like the water treatment plants? Yeah, so a lot of wastewater treatment plants actually are a source of sodium and chloride. So they add sodium and chloride during the wastewater treatment plant process, and that can further exacerbate the problem. So it's not being removed, and there's also a little bit being added during the wastewater treatment plant process.
Good question and good answer. One more question right here.
Yeah, is there a neutralizing agent that we could potentially use to counteract this, or is that just adding more chemicals and not a good solution, or should we just add more snow days?
I think snow days is the answer to that, but I'm actually not sure if there's a neutralizing agent. That would be a good thing to look into. I know a lot of places are switching to using brines on their roads instead of just road salt, because that actually lowers the concentration of salt that they're applying to their roads. So that could be a possible solution and something that people are moving towards in the future.
Fantastic. Well, thank you so much.
Thank you.
All right. Thank you very much, Professor Obermeyer. So just wanted to paint a quick scenario. The holidays are coming up. Maybe you're taking a car trip to visit some family. And along the way, you want to hear some Christmas music on the radio. Wouldn't it be great if there was a mobile app that just based on your location told you what stations you could hear? It seems like a really simple concept, but there really isn't anything as far as a mobile app that does that you can download on your phone. And that's always bothered me. So enter the culmination of my research, Radioland, where I attempted to build a mobile app that based on any location in the US or Canada told you what FM radio stations you ought to be able to hear. So the first thing I had to find out was this even possible to predict signal strengths of FM stations? There's several different factors that can affect how a signal is received by your radio, if there's any hills or mountains in between where the station's transmitting from. So we were able to make contact with a wonderful engineer at the FCC, the Federal Communications Commission, and we got access to a study that was conducted back in the 60s that looks at how signals travel between two places. We supplemented that with terrain data, again to account for hills and mountains, and I put together a database of over 22,000 FM stations in the US and Canada, and that's linked with Radioland. So Radioland either uses your device's location, or you can search for any location in US or Canada, and you get back this handy list of stations that shows each programming format for the station. This little signal bar indicator, similar to what you might see on your cell phone, that'll depict how strong the signal is. You can filter your results. If you just want to see Christmas music stations, you can type that in. And then for our more technically savvy users, we let you visualize where you're at in relation to where the station is and the terrain in between, so you can see if there's any hills or mountains blocking the signal. My early results, as far as accuracy, have been very favorable in locations where there are terrain differences, like the Poconos or maybe the Rocky Mountains west of Denver. And I'm really excited to say that we're looking to release Radioland for Android and iOS this winter. If you log on to radiolandapp.com, you can submit your email, and you'll be the first to know when it goes live. So on your next family trip, you'll know what's on your dial. I'll take any questions. Thank you.
Wow, that is fantastic. And you did all of this over, what, the last year? How long has this project taken?
Since the research started in the summer, and it's really been kind of overdrive this fall, basically. Just
Great.
getting it.
Has anyone ever told you you have a voice for radio?
Practice a little bit.
Questions for Nick? Yes, right here. Christopher.
Thanks. Do you see this helping college radio stations get more exposure in some way, shape, or form?
Absolutely. I think, you know, I don't want to say radio has been forgotten a little bit, but, you know, there's a lot of streaming options. That's something we had to weigh when we were developing this app. So, yes, I think to bring, you know, just letting people know what's around, and all this, again, is free. You know, it doesn't cost a subscription fee. And I could definitely, you know, we can specifically highlight college stations like Villanova's radio station. So I think it would be a great way to get it to people. Great question.
You know what? I think there's a networking opportunity. User interface, app development. You guys should talk.
Always looking for testers. That's,
One last question, right back here.
I actually just have a comment. I worked with Nick when he was in the College of Professional Studies, and I just appreciate all you've done for Villanova. And he's always made connections, and just the advancement that I've seen. I'm so proud of you.
Yeah.
So congratulations.
Have you ever wondered how our cells respond to different kind of stresses like nutrient scarcity or damaging environmental toxins? So to simplify the whole process, whenever a cell receives a stress signal, that signal is processed in a series of molecular events in the cell, which is called signal transduction. And the process of signal transduction eventually results in the response from the cell. So usually the response from the cell is in the form of gene expression. So it is really important that genes are only expressed when they are needed. So the whole process of gene expression is controlled by these proteins called transcription factors. So the way transcription factors work is they have a binding site in the DNA where they bind to, and from there they can recruit different kind of machinery to either repress or activate the expression of the gene. And genes, as we know, code for proteins, and proteins virtually do almost all cellular processes in our body. That brings us to my research. So I work with the pathogenic yeast Candida glabrata, which causes candidiasis in humans. And Candida glabrata is actually closely related to Saccharomyces cerevisiae, which we commonly know as the baker's yeast or the brewer's yeast. So just like humans require thiamine or vitamin B1 to survive, Candida glabrata also requires thiamine to metabolize carbohydrates and amino acids. When the going gets tough and there is no thiamine around, it has a set of genes that it can upregulate in a process called thiamine signal transduction pathway, and the genes are called Thi genes. So the expression or the activation of Thi genes is under the control of interaction between these two proteins, Thi3 and PDC2. So when you have high thiamine in the cell, there is no interaction between these two proteins because you don't need Thi genes to be activated because you already have high thiamine in the cell. But whenever there is low thiamine in the cell, you need these two proteins to kind of interact with each other and activate those genes so that they can help you survive, basically. So I specifically work with PDC2. So to understand the whole process of thiamine signal transduction, it's really important to understand how this interaction between Thi3 and PDC2 occurs and where exactly in the DNA does PDC2 binds to. So over the summer, so PDC2 protein has different regions, right? So it has a DNA binding region that binds to the DNA. It has an activation domain that interacts with Thi3 and activates the Thi genes. So over the summer, I was able to show that the DNA binding domain or the activation domain by themselves are not enough to bind to Thi3 and upregulate Thi genes. And right now, I'm in the process of optimizing this protocol that will allow us to get more insight into where exactly in the DNA does PDC2 binds to. And you might be wondering why studying a random protein in a random yeast is important. But according to a study by CDC, in 2017, the total cost that was bared by Americans who had candidiasis was $3 billion. And this amount is expected to rise because Candida, it turns out, are really good at evading the antifungal drugs that we have because of antifungal resistance. So if we can study this Thi pathway, and if we can understand how this interaction occurs and where exactly in the DNA PDC2 is binding to, we can potentially figure out a different drug target that we can then use to create antifungals. Yeah, that is my, thank you. I'll take any questions.
Kendra, thank you so much for that great presentation. Questions for Kendra? Kendra also sits on our Graduate Student Council, so any graduate student needs for Kendra? Yes.
Do you think that this sorry. I was just going to shout. Do you think that this pathway is conserved with other organisms?
Yes, actually, we have a pretty good understanding of how this pathway works in Saccharomyces cerevisiae, and we know that Candida glabrata is missing some of the elements of this pathway, so that is why it's more important, because we already know how it works in Saccharomyces cerevisiae, but we don't know how exactly it works in Candida glabrata, so that's one of the focus of my study, to kind of map out the whole pathway. And it also helps that Saccharomyces cerevisiae is a model yeast, so everything kind of goes from there.
Other questions for Kendra?
Thank you
so much.
Thank you.
I'd like you to think of a conspiracy theory. You might be able to think of several just off the top of your head. Some might seem relatively harmless, like aliens in Roswell, New Mexico, but others might seem more sinister, like the QAnon conspiracy theorists who stormed the Capitol on January 6, 2021. Conspiracy theories have entered the popular culture of the United States, and no matter how ridiculous they may seem, they can be incredibly dangerous, especially when mixed with politics. But the intersection of conspiracy theories and politics is not a new phenomenon in the United States. Historians have long known that conspiracy theories played a part in American politics as far back as the colonial era and the American Revolution. Yet historical inquiry into political conspiracy theories often seeks to understand why otherwise rational people believe seemingly irrational explanations of events. This interpretation seeks to answer the question of why conspiracy theories exist, while also aimed at showing how dangerous conspiracy theories can be to civil liberties and democratic government. This line is problematic, as it makes many conspiracy theories seem like a natural occurrence stemming from religious, social, and cultural aspects of the American experience, such as the fear of powerful individuals or just a belief in fairness. I took a different approach in my research to reevaluate the role of conspiracy theories in the politics of the United States. Instead of asking why people believe in conspiracy theories or where do conspiracy theories come from, I asked how are they spread and by whom, and then I asked the question why. My research found that conspiracy theories and conspiratorial rhetoric are not just part of a cultural script that Americans draw on, but that they are a political technology, a refined and innovatively used tactic of political actors to gain power. In my research, I looked at the ways that early to mid 19th century politicians in the North used conspiracy theories to gain power, influence public opinion, and therefore win elections. Using the letters they wrote and the newspapers they printed, I found that the same politicians returned to the tactic of using conspiracy theories over the course of several decades, simply changing the object of their conspiratorial rhetoric from groups like the Freemasons to Southern enslavers. Northern politicians used conspiracy theories most often when they needed the support to build new political parties or to better define the platforms of existing ones. The reason for this was that conspiratorial rhetoric gave them a means to appeal to the exclusively white male voter base. In short, the reason that conspiracy theories were so successful was that they were employed to reinforce white supremacist and patriarchal conceptions of democracy and American society. Thank you.
Thanks very much, Chris. I have no idea why I hate that hate on Uncle Sam. I actually don't mind Uncle Sam. For some reason, I made a connection to the IRS, and I was like, yeah, boo hiss, IRS.
Conspiracy theory.
Yeah, yes. Conspiracy. I mean, I may be a little partial to Rosie the Riveter, but, you know, Uncle Sam's okay, too. Any questions for Chris? Yes, Kendra.
I have a question. What actually defines conspiracy theory? Because, like, as time progresses, we know that there actually have been things that were previously labeled conspiracy theories, but turned out to be true because I guess the government was withholding, files and stuff like that. So how do you exactly define them?
So I would define conspiracy theories as any kind of idea or narrative that an individual or an organization kind of works to gain power at the expense of others behind the scenes. And part of my interest in conspiracy theories is that they don't necessarily have to be true or false. One conspiracy theory I focused on was called the slave power conspiracy, which was held by Republicans before the Civil War. And they argued that Southern enslavers were conspiring to take over the American government through holding the presidency and various, postings in the federal government. That actually turned out to be more true because many Southerners did, and they had a vested interest in preserving slavery by holding positions in the federal government. But what's important is the way that Northerners and oppositional politicians employed these to win voters and to influence elections by riling people up against these perceived conspiracies, whether they were true or false.
Depression is the leading cause of global disability, with an estimated 322 million cases worldwide. Unfortunately, though, even the most effective treatments have response rates of only about 40 to 60%. This means that for up to 120 million people, current depression treatments aren't working. Furthermore, for this population in which treatments are not effective, the genetic and biological factors that contribute to this lack of response, also described as treatment resistance, are still largely unknown. Something that has been theorized to contribute to some forms of treatment resistance is low levels of the neurotransmitter serotonin. Millions of people are taking antidepressants to try to alleviate their depression symptoms, but many individuals do not have access to or choose not to utilize medication approaches. One alternative low-cost approach that shows promise in alleviating some symptoms is cardiovascular exercise. My project is concerned with understanding how low levels of serotonin impact exercise interventions intended to reduce depression symptoms. Specifically, my overarching research question is, does low serotonin prevent the antidepressant-like effects of exercise? To examine this question, I utilized a mouse model of serotonin deficiency. Exercise is known to reduce depression symptoms in some individuals, but whether genetic factors like serotonin deficiency dictate an individual's sensitivity to these benefits remains unknown. One mechanism that is thought to contribute to the antidepressant effects of exercise is increasing hippocampal neurogenesis. Hippocampal neurogenesis is the formation of new neurons in the hippocampus, a structure within the medial temporal lobe of the brain. This study compared wild-type mice with 60 to 80% less brain serotonin, who either completed 4 weeks of wheel running exercise or served as non-exercise controls. At the end of the study period, these groups were compared on several anxiety and depression-like behavioral measures. I found that on half of these measures, the low serotonin mice did benefit from exercise, but not as much as the other mice did. One of these measures, on which I saw significant effects, has previously been reported to require hippocampal neurogenesis. For the next phase of this research, I will complete cellular quantification of neurogenesis in these mice to further understand its impact on the behavioral results I saw. Overall, the findings of this study suggest that exercise still appears to lead to some antidepressant and anxiety reduction benefits in serotonin-deficient animals. In the future, I'm interested in learning more about why these mice don't respond as fully to exercise and how we can boost their response, as this has significant implications for depression treatment response. Thank you.
Thank you so much, Allison. Questions for Allison? Yes, right here.
Does these low serotonin mice, do they respond to antidepressants like SSRIs and those kind of things as well?
No, but there's an expert back there who can tell you even more about that, but the short answer is no.
Yes, right here.
How do you find mice with, do you look for mice with lower levels of serotonin, or do you treat them with something to get rid of the serotonin?
These are genetically engineered mice in the lab, so they're bred to have this reduced serotonin.
Yes, Kendra.
Yes, what were the parameters you were comparing?
There are several behavioral measures. I can tell you the names of them. I don't know if it would be meaningful, but I'd be happy to. It was the novelty suppressed feeding test, the open field test, the elevated plus maze, and the forced swim test.
All right, fantastic. Thank you.
So climate change is ever present in our collective conscious today. We're thinking more about carbon more than we ever have before. And in general, we think of trees and forests as our carbon banks where carbon is stored. But the real powerhouses of soil carbon storage are peatlands. And peatlands are a type of wetland that is characterized by a dense soil organic matter that is called peat. It's very mucky, and I can tell you that from personal experience that makes them not very fun to walk through. But it does allow them to hold an astounding 500 billion metric tons of carbon worldwide. For context, that is more than the US has emitted through burning fossil fuels since the invention of the steam engine. Now, peatlands are able to do this in part because of local cool and wet conditions that prevent peat from decaying rapidly. But we're concerned that as climate change drives the warming and drying of peatlands, these systems will no longer be able to accumulate carbon well and will instead begin releasing it into the atmosphere, which will amplify the greenhouse effect. Now, peatlands are understudied in many ways, and this is particularly true in temperate regions, such as here in the eastern US. And my research aims to fill in some of these gaps in our knowledge of peatlands. Specifically, my aims are to characterize the plant communities of Pennsylvanian peatlands and to relate these communities to environmental variables such as how much shade is available or what nutrients are in the water. Now, this is important because different plant communities have different relationships with the carbon cycle, and because in order to properly conserve peatlands, we need to fully understand them. So to date, I have gathered samples from 28 microhabitats within 3 peatlands in the Pocono Mountains, and I'm currently in the process of analyzing these samples to gather my raw data. And once this process is complete, then I will input my data into a statistical model that groups the communities based on their relative similarities to one another. The model also compares these groupings to the environmental variables that I've analyzed to determine which ones are most important in peatland plant diversity. Finally, I will produce geospatial models that will show clearly to anyone what my results are through maps. My work also promises to have strong real-world impacts because I have collaborated with scientists at the Pennsylvania Natural Heritage Program throughout my work, and I will share with them my results so that they can use these directly to benefit peatland conservation strategies here in our home state. I strongly believe that understanding and conserving peatlands is a critical step to ameliorating our climate crisis and guiding us into a brighter, greener future. Thank you.
All right, questions for Joshua? Yes, right here.
It would be a very silly question, but is it possible to create peatlands? Like if there were like lands that were left over from like old strip mines or something, could you plant a mixture of plants to create like an artificial engineered peatland environment?
The answer is yes, but it would not be very efficient, and that's because peatlands accumulate all this carbon over thousands of years. They actually don't have very high productivity. They just have such slow decay that they're holding on to thousands to 10,000s of years worth of carbon.
One more question.
Do peatlands exist all over the world, or is it unique to North America?
So peatlands are concentrated in the boreal zone, which is just the subarctic zone, so about 50 to 70 degrees north. And there are, to a lesser extent, also in the temperate zones around the world. And there are very few in the global south, particularly in Argentina.
Fantastic. Thank you, Josh. All right, that concludes our presentation part of our research symposium. I hope that you enjoyed hearing the diversity of research that folks are engaging throughout the college. We learned about peatlands. We learned about policing in Philadelphia. We learned about user interfaces and exciting new apps. This is, again, just a small sample of just the wonderful work that our graduate students are doing. So please join me in just congratulating all of our presenters today and all the fine work by our graduate students in the College of Liberal Arts and Sciences.